My name is Anotida Madzvamuse. I'm professor of mathematics in the Department of Mathematics here at the University of British Columbia. My research focuses on understanding single cell dynamics, how single cells navigate through complex environments. My goal is really to try to develop an artificial cell, a synthetic cell, that we can carry out experiments computationally without the need of involving animals or expensive costs. You have a primary cancer, for example. In many cases, physicians, once they identify it, they can take it out, depending where it is. But it turns out, in many cases, that depending on the cancer, a few months later, or even a year later, they have problems somewhere else. They have removed this tumor, primary tumor, but now there are other secondary tumors which are forming somewhere else. How do secondary tumors form from primary? And one aspect of this is these single cells that are migrating from a primary tumor where they are concentrated together to form the tumor. They live individually as individual cells, and they go into the bloodstream. They are transported by the bloodstream to other areas of the body. And once they are there, they leave the bloodstream through the tissue again, the veins, and they go and settle and start to form again these secondary tumors, which are very difficult to detect. We only see them much later on. So when cells migrate, they find different tunnels, for example, and they explore these tunnels and see which one is optimal for them to go through. How much force do they need to pull themselves through these complex environments? What I'm looking for is really the geometry and the dynamics, both inside the cell and outside the cell. How these drive the decision making of cells. How do they make these decisions? It's part of us trying to translate these observations which come from experiments into mathematics.